Hi everyone, my name is Savanna. Thank you so much for watching my presentation. Unfortunately, I couldn't make it to class today, but this is my presentation on salamander mussel eDNA sampling sites. So so if you're unaware, the salamander mussel is the mussel that I study in my research. It is endangered but native to Illinois and often a target for conservation. And we are using eDNA instead of conventional sampling methods to try to find populations throughout the state. And often we have to decide where we want to conduct eDNA sampling because the locations of current populations are unknown. So to decide where we want to do eDNA sampling, we have to think about behavior, natural history of the organism, and we also have to think of abiotic conditions as well, such as UV, water velocity, water temperature. So that's what I wanted to do in this project today, and use geostatistics to do that. So there's two parts to my project. One being I wanted to find bridges across the Sangamon River in Champaign County to hopefully assess where potential sampling sites could be for myself. And then I also want to look across the greater Illinois River watershed to look at water quality attributes to make a weighted model. So moving on into part one, we're going to be looking at water velocity and eDNA collection. So, like I said, water velocity is a abiotic factor that does influence eDNA collection. And fortunately for this specific species, a model has been developed to understand the relationship between water velocity and eDNA concentration. So first, I wanted to identify bridges with the National Transportation Data Set with shapefile point data. And I did this because they often act as artificial habitats for mussels because of the concrete rock formations that happen during construction. I went into R and I coded based on the model by Porohanes et al. In 2023, and I used the bridge data and then water velocity data from the National Hydrography dataset. So here is my first map and chart. I wanted to specifically look at this chart. We see that on the X there is water velocity, and on the Y, final eDNA concentration 10 kilometers downstream from the bridge. And as we see, there's a very clear linear relationship there. And we can actually, so like this top point here has a really high velocity at 1.75 kilometers per hour, and then therefore has a high eDNA concentration. So based on these, I looked at a map and I symbolized it in a way to show that the darkest red are the best sampling sites. So for example, here's one of my current sampling sites right here. It's sorry, there we go. It has a pretty medium velocity, therefore a pretty medium concentration, and so maybe looking at more of the red dots. So that's what this map is showing. These are all the bridges in the Sangamon River in Champaign County, and here are the five I want to specifically look at. So I made a flowchart to kind of see where these sites would be and to see, oh no, it froze. Okay, but you get the gist. It goes through when it's not frozen. So then here's the general scheme of field work in my lab. So go out. Well, first we do our genomic preparation, and then we head out and sample. And here's a picture of me sampling at one of my previous sites. So moving into part two, we looked at watershed water quality, and this is the watershed I looked at. It's hydraulic code 0713. Here is that in a map. As we see, this is the Illinois River in this dark blue right here. So I wanted to make a weighted model with water quality attribute data from the National Hydrography data set based on specific variables and how they relate to the preferred habitat of the salamander mussel. So first is minimum elevation. Mussels prefer lower elevation because that's where their hosts are. Specifically, the host of the salamander mussel is the mud puppy. So I weighted that so that lower elevations have a code of 1, and we see the Illinois River kind of follows that trend. Next is slope, really similar to elevation. We have lower slope is preferred, often because it's easier to latch onto rocks with lower slope. Here is runoff. Mussels prefer lower runoff because often with high runoff, there's high nutrient load, so that could be overwhelming for the mussel. And here is flow. High flow is required for the filter feeders to actually obtain their food. And, oh, I skipped one. And here's water velocity. I already covered this, but again, velocity is required to bring in as much food as possible for the organisms. So with all that combined, I did a hotspot analysis, and this is the map that shows that. So cold spots are the darker blue ones, and they indicate clustering of lower values, which is actually what we want, because the lower values are what I designed to be better habitat quality. And on the opposite spectrum, red spots are the hot spots, and they are potentially worse quality. So funny enough, where I sample right now is pretty red. So maybe I'd be better off finding populations in the Illinois River and more in southern Illinois. So looking at the bigger picture, connecting back to conservation, of course, this is a rare endangered species, and a lot of the populations are unknown across the state. It was actually considered to be extinct for a bit, and then we were able to find some. So just trying to really pinpoint with evidence-based data where we can do eDNA sampling, and I'm so happy that we were able to do geostatistical analysis to find that. And then just having more efficient sampling can result in more conservation and more money attributed to conservation down the road. And here are my references. Thank you so much for watching, and I appreciate any feedback. Thank you.